Hello, everyone, and welcome back to the final lecture for this week. This is going over Chapter 9, Sections 5 and 6. This will be about the visual brain in action and the plasticity in the visual pathways. So anatomical and physiological studies of the brain systems, they leave one critical question unanswered. How do all of the cells in these systems act together to produce a particular function? One way to answer this question is to evaluate what happens when dysfunctional parts of the visual system take place. So let's imagine there's an injury to the pathway leading to the cortex that could lead to monocular blindness. Now, monocular blindness, that would be the destruction of the retina or the optic nerve in one eye, producing loss of sight in that eye. Well, that makes sense as we think about this from an anatomical perspective, right? So the retina, that is the part of the eye that's on the inside where the light is being projected to, if we think back to that lecture. So if there's damage to that projection stream, like if you're trying to use one of those projectors to watch a movie and you have the whole screen pulled out, if that gets damaged or torn up, then even though the light may be going back there, if it's not getting projected onto anything, you're not going to be able to really watch the movie or experience it. Similarly, remember that optic nerve, that's what's carrying all the information from all the photoreceptors to the brain from the eye. So then all of the information from that eye is not being brought over. Now, this is an important distinction from homonymous hemianopia. This is blindness of the entire left or right visual field. Now, again, let's make this distinction between what these two conditions might be from a clinical perspective, right? If somebody has damage to an eye, that means that all of the information from one eye, some from the left visual field, some from the right visual field, no matter which eye it is, some of that will be lost. Here in homonymous hemianopia, this is blindness of an entire visual field. So although both eyes may be working from the retina and the optic nerve perspective, there may be damage further down the line in the system where the information on one specific side of the visual field is not being coded or passed along. Then there is quadrantinopia. This is blindness of one quadrant of the visual field. So there's the left and there's the right visual field, but then we can also split that horizontally as well. So there's the upper quadrants and the lower quadrants. And that's part of the reason why you may see in some like hospital shows like Grey's Anatomy and whatnot, if they're ever doing testing of visual fields, they will check, they'll go to the four corners too, because some people, it may be that they can see things on the left and the right, but if you start to go upper left, they cannot see it because there's one quadrant where they're having difficulty seeing things. Then there is scotoma. This is a small blind spot in the visual field that's caused by a small lesion or migraines of the visual cortex. Now, some of the consequences of lesions in region V1, that primary visual cortex. We can see some really nice examples here of what we were just talking about. So in the case of a homonymous hemianopia, like in image a complete lesion of V1 in the left hemisphere would result in a hemianopia affecting the entire right visual field. So we can imagine this person, they're not seeing functionally any information on this side. This is all they see in the world that's around them. Despite that all of the light is still hitting their eyes, the damage to this part of the primary visual cortex means that they are only able to functionally see the left side. Now, in part B, if they have a quadranopia, a large lesion in the lower lip of the calcarine fissure that may produce this quadranopia that affects most of the upper right visual quadrant. So here they're still getting the information. They've got some on the right and even maybe some in the upper right quadrant because there's not magic to it. It's not like there's these nice clean lines for a lot of people, but there are these larger blind sections where they may not be seeing anything. Then, and that's why in some of the testing that can be done from this functionally, we can ask people to draw pictures or copy images that are shown to them. And what's really interesting is some people, they may totally ignore a certain section, and it's not really that they're ignoring it, it's that they don't see this one particular area of the image that we're showing them. Then in image C, for individuals who may have had scotoma, this is that smaller lesion in the lower lip of the calcarine fissure. That can result in even this smaller blind section. And that's one where it may be easier or it may be more difficult to actually catch this because as a person is just moving their eyes around, they're scanning their surroundings, they're able to fill in the space as they're just moving their eyes around. Now, what about an injury to the ventral stream, that what pathway? That can lead to some really interesting pathology as well. So one term or experience is this thing called agnosia, which is just a not knowing what something is. And there's a couple forms that this may take. So there can be this visual form agnosia, which is an inability to recognize objects or drawings of objects. There can be a color agnosia, which is an inability to recognize colors. And we talked about this before, but there can be a face agnosia, which is an inability to recognize faces. Now we have this example here of this patient who came in, JW, and this patient survived a severe heart attack while exercising. As a result, he suffered anoxia, which means he wasn't able to get oxygen for an extended period of time. And this caused damage in the occipital region of his brain. As a result, he couldn't recognize simple line drawings when he was shown them. So this was kind of like what I was talking about a moment ago. Here, the doctor showed JW this picture of a table. He was asked just to copy it. He's still seeing this image while he's trying to draw this, and this is what he's producing. We can see this pretty clear distinction here. Now, it's not like he's totally guessing. He's got this rectangle type figure, right? He knows that's going on. So it's not like he doesn't have anything, but it's clear that this isn't somebody who's drawing a table. Here as well, it's interesting because we have this square and triangle. He's able to produce, he has a square, he has a triangle, but he's switching the sides of where they are. And the orientation, he made it one figure. It looks like he maybe didn't even pick up his pencil the entire time he was drawing. And again, this is interesting because it's not as though we're removing this. He's not going off of memory. This is him copying down what he's seeing. And finally with this image as well. And in this case, he's able to copy kind of the circle type design, but he's not able to include the other pentagon. What about an injury to the Howe pathway? Well, that can result in something called an optic ataxia, which is a deficit in the visual control of reaching and other movements. This can result from damage to the parietal cortex, but there does seem to be a retention of the ability to recognize objects normally. So essentially, people with damage to the parietal cortex in the dorsal visual stream, they can see well, but they cannot accurately guide their movements based on visual information. And this relationship illustrates that the dorsal stream functions to guide movement. In contrast, people who damage the ventral stream, they cannot perceive objects because the object perception, that's that whole ventral stream function. But these same people, they can guide their movements to objects based on visual information that they have. So here we have this other example of a patient, and in this case, each line passes through the points where the index finger and thumb first contacted the perimeter of the shape on individual trials in which the subjects and the controls were instructed to pick up the shapes. So DF isn't able to recognize these shapes, not able to do it. But she and SH, the control, this is a person who's entirely healthy, they both place their finger and thumb appropriately opposed points on either side of the shape each time, consistently. Meanwhile, here we have RV, whose object recognition is impaired. They're taking these unstable grasp points that often don't pass through the object's clear center of mass. So they're not able to control some of that functioning that they're doing to pick the area, the object up in an area that would be most effective for them. So what about the plasticity in the visual pathways? Although the anatomical organization of visual pathways reflects the unfolding of a genetic blueprint, the visual system is really quite plastic. The manipulation of visual experiences during prenatal and early development and beyond, it can profoundly affect our visual perception. People can show remarkable plasticity after injury to and even complete loss of the visual pathways. An example is the ability of blind individuals to echolocate, which is correlated with the emergence of auditory spatial maps in the occipital cortex. So there is this manipulation of visual experience in the young brain, which can chronically alter visual perceptions. Injuries can also prompt these radical changes to the sensory systems. So individuals who have had strokes involving the occipital cortex often have severe visual impairments immediately after the event, but they tend to show significant recovery over the ensuing weeks or months as the brain changes to accommodate this injury. And these individuals often respond well to rehabilitation, even when initiated long after the injury, which is really great news. It's really exciting to see. Now, an injury to the occipital cortex early in life, that can result in extensive rewiring of the visual pathways. A genetic enzyme disorder, which resulted in a bilateral occipital lobe injury in the first 2 weeks of this one patient VI's life, it took place. And when they went back and looked at BI at age 6, they found that despite extensive occipital lobe damage, BI still had surprisingly good conscious visual abilities. And an object that is represented by a visual network of many cells within TE and beyond, our visual abilities are not solely genetically determined, but they're modified significantly by our experience. And this fact is actually in the visual system and our other sensory systems as well, which is, again, just pretty amazing to see. And I think some really good news about some of the neuroscience there. That is the end of this week's lectures. Make sure you do end up completing all of the other tasks, quizzes, and psych sims for this week, as well as the weekly quiz. And best of luck on exam 2. Bye-bye.